Once again. So let's dive right into the next topic, which is the evaluation of bacterial colony growth. So as we have been talking from the previous discussion, right after being able to inoculate the sample, incubating it for 24 hours using the right incubation technique, then we can and selecting, okay, so first pala, let's backtrack for a bit. After receiving a properly collected sample, you select the appropriate culture media for that specific sample or for what um, bacteria are we expecting to culture based on the symptoms of the patient. After that, we're going to be inoculating it or streaking it properly again um, using a very good aseptic technique. After that, we're going to be incubating it properly once again, either aerobically, anaerobically, or in a capnophilic environment or microaerophilic environment. And after we've all ensured those things, then we can expect growth. Okay? We can expect colony growth within the culture media that we have inoculated or we have stripped. Okay, so what is now the next step after we have successfully isolated or we have successfully grown or cultivated the organism? So of course, the next step is for us to evaluate the growth. Kasi again, what do we want from this, or what would be one of our main goal is again to identify the organism. So all of these things or all of the methods that we've been learning so far, except for the inoculation, would be procedures that will help us give or yeah, that will give us clue as to what will be the identity of the organism. So from a gram stain, from ano ba? From the gram stain, yes, and then some of the staining techniques that we have recently learned, ano ba, mga stain for the flagella, stain for the endospore, ganyan, ganyan. Again, these will provide us with more clues. And of course, their growth would also give us very valuable information as to what would be the most likely identity of the organism. Now, there are some organisms who have very specific or characteristically unique growth, okay? And that's very beneficial since it will give us a very definitive identity. Okay? But not all has that characteristic as we will be learning later. So before we go along with our topic. So what will be the learning objectives? First is for us to examine and evaluate the growth of bacteria after incubation. And how are we going to be able to do that? Of course, by describing the colony morphology using limited or accepted descriptive terms, and also for us to compare the growth on solid and liquid culture media. Um, kasi magkaiba yung composition since there's a difference in the composition of the culture media. First, we can expect a different type of growth on both of these medium. Okay, another thing. So if we have a sterility checkpoint when we prepared the culture media, although it's not usually done, to be quite honest, you also have a sterility check on a prepared or on an inoculated or yeah, in a grown na bacteria. So how can you check if uh diba, if hindi contaminated siya or hindi? One thing is you can actually perform a gram stain and check if there are multiple colonies. Okay, so that kasi as much as possible class you want to isolate pure colonies. Although there are some instances wherein dalawa yung you're harboring, the patient is harboring two organisms, but that is usually more common on blood samples. And the uh, the uh, the machine can actually detect that. Okay, so but if it's on a non-sterile body fluid, lalo na kapag urine, mga sputum, if there are multiple or if there are present multiple organisms, like for example, your gram stain and then there's gram negative organism, and then you are also be able to observe multiple shapes, and the gram negative rods, gram positive rods, gram positive coca, and then na lahat, then that is most likely an indication that that is contaminated. So that's how we can check. And you can also notice it on the actual growth, no? Macroscopically using a naked eye, you're going to be able to observe what do you call that? Talagang magkakaiba yung itsura. Like, iba yung color ng ganito, iba yung size, iba yung uh, diba, arrangement, mga ganun. And most of the time, ang common kasi na contaminant is fungi actually. So you're going to be observe some moldy or fluffy growth. And again, those are very indicative of fungal contamination. Since madali lang kasi ma-spread ang fungi, diba? Kasi they have spores. So it's a very common contaminant in the bacteriology section. Okay, now let's go over the evolution of colonies. So if maganda yung growth, okay, you were able to observe a very uniform growth, then we can go right ahead in the evaluation. So we can evaluate a culture media or the growth of a colony through relative quantity. Meron din naman tayo yung talagang quantitative, pero iba yung technique nun, guys. That's usually using pour plate method wherein you're not going to be streaking it. Ipo-pour mo lang yung sample mo alongside the agar before it's going to solidify. That's called a pour plate method. Instead of you streaking the sample, what you're going to be doing is after mong mailagay or dun sa petri dish, maglalagay ka ng sample. Usually kasi water, this is for water analysis. Or in dairy, ganun, kapag liquid yung sample mo, you can um, put a very specific amount in a petri dish, saka mo i-spread doon yung, or ipo-pour yung culture media. So it's called a pour plate method. And yun, you're going, for that, you can actually count the colonies. Okay? You can count the colonies, lang isa-isa, using a colony counter. So it's like it's a machine, an equipment that will help you. May mga grid kasi that will help you count the colonies better. Siyempre kasi if you're going to manually count, you can, but it's very hard to be accurate with that kasi again, the colonies are very small. So we can use a quantitative method using a colony counter for a pour plate method. And you can also count that even if you used how do you call that? Even if you use a streaking method, and that usually applies to a urine sample. Kasi for urine sample test, if you went over the PowerPoint for the inoculation techniques, you may observe another type of streaking method. We call that the continuous streaking method. So medyo mas madali siya kasi. Ganyan. May ganun ka lang, one straight line at the middle, and then you're going to be streaking it all around or across the agar plate. So we call that the continuous streaking method. So for some, there are some iterations wherein meron din siyang specific pattern, but generally, that is the idea when it comes to a continuous streak, and that is usually used for urine sample. Why? Or kasi, para ma-quantitate natin yung amount of organism so that we can count it. Kasi kung gagamitin natin yung quadrant technique, di ba, makakapag-count lang tayo ng mas maayos on the actual third and fourth quadrant. And mas maganda yung continuous streak, again, for urine, because we want to be able to quantitate it. Kasi, in order for a urine culture to be considered significant, it should have at least 100,000 colony-forming units per ml. Again, take note, in order for urine cultures to be significant, meaning, there could be a potential bacterial infection within the urinary tract, it should have 100,000 colony forming units per ml quantity of bacteria. Kasi again, diba, once it goes through the urinary system, particularly the lower part of the urinary system, hindi na kasi sterile yun. So, it's normal na may small amount of bacteria within the urine. So, yung misconception na sterile daw urine, it's not the case. Lalo na if it has been stored within bladder for a longer period of time. So it makes sense that there are some very small amount of bacteria that could be present there. But if it's more than 100,000, then that is already a significant finding, or it's a significant amount of organism that may already cause an infection. So that is for the actual quantity, but we can also use the relative quantity method. So this you can use for the four quadrants trick, wherein you're going to be getting the approximate number of colonies per quadrant. And then you're going to be giving a score. So if there's less than 10 for the quadrant, tapos wala on the other one, and that's 1 plus, and so on and so forth. So this is a general guideline, and individual laboratories may vary in the methods used for quantitation. Now let's, by the way, this is not usually done anymore. Kasi again, laboratories usually use or opt for the use of automated machines, kaya hindi na siya masyadong ginagamit. Okay, how about for colony characteristics? So aside from the number of colonies that have grown, of course, you need to take into account how they look like or what they look like, and these are referred to as colony characteristics. So noting key features of a bacterial colony is a very important technique in order for bacterial identification, as the success or failure of it often depends on the accuracy of this observation. So, why is it important, class, that we, evaluate it properly? Because again, that can correspond or directly correspond or correlate to what would be the identity of the organism. And by observing the colony characteristic, the microbiologist is able to make an educated guess regarding the identification of the isolate. Diba? Just like us, human beings, diba? There are certain characteristics that are unique to only us, and that is also applicable to organism. Therefore, when we characterize or describe their growth, then again that uh, helps us go nearer and nearer towards the identity of the organism. So one important factor, one important consideration in the evaluation of colony is the size. Okay, so uh, this is not usually or doesn't give us much information, but it can give us a good way of comparing very similar species. So as the first bullet says, colonies are described as large, medium, small, or pinpoint. So ano na yung general description. And size is generally a visual comparison between genera or species. Again, it doesn't give us much vital information, but it can be very useful in comparing or differentiating between genera or species. So a very good example of this would be between Staphylococcus and Streptococcus. So generally, they both have very similar colony growth. So medyo whitish or creamy yung colonies nila. But one thing we can use to describe them is their size. Okay? So see, Streptococcus are very small. Okay? Streptococcus are very small, and the Staphylococcus are larger. Okay? So Staphylococcus are larger, and Streptococcus colony are smaller. Kaya pinpoint, diba? Pinpoint yung tawag. Tapos pinhead naman yung sa Staphylococcus. So aside from size, you can also evaluate them through form or margin. So the edge, you say margin or form, the edge of the colonies should be observed, and the form or margin is described as, or are described as, smooth, filamentous, rough, or rhizoid, or irregular. So just like what the image is illustrating, or what the illustration is showing. So again, very easy. For filamentous, of course, it's branching. Okay, it has filament-like structure. For smooth, you can clearly see a rounded colony, and for rough, and medyo parang may mga broken lines. Hindi siya complete. Kasi again, we're talking about the margin or the edge. Okay? So if hindi completely smooth na round yung colony, then most likely that is a rough margin. And last, irregular. Okay? So if the colony grew a very odd pattern Of course, the margin will follow that shape, and we describe that as irregular. So colonies of Bacillus anthracis are described as medusa heads because of the filamentous appearance. So microbiologists describe Bacillus anthracis as having a medusa head growth or margin kasi very filamentous or maraming filament that looks like snake. And that's why it is referred to as medusa head. Pero hindi naman siya all the time ganito yung itsura. But if you um, see a medusa head appearance or very filamentous appearance, then that is a good indication that it may be Bacillus anthracis. So, you can observe a lot of this class na medyo malawak ang imagination ng mga microbiologists natin kasi when it comes to describing colonies, madami tayong name for them. It's like, for example, medusa head colonies for Bacillus anthracis. Okay, for the next one, Proteus species also exhibit a very unique phenomenon referred to as the swarming phenomenon. So as you can see from figure 8.9, this is a swarming, or this is the swarming of colonies of Proteus. Again, all Proteus species are capable of exhibiting these phenomenon or this growth. Okay, nagsuswarm sila. So parang veil-like appearance, yun din yung minsan na ginagamit na term. Veil-like appearance all throughout the agar or swarming phenomenon. Again, don't forget, that is a very specific characteristic of the species of Proteus. Also, diphtherioids. Mga diphtherioids, just like Corynebacterium diphtheriae, have very wrinkled colonies, okay? Very wrinkly colonies. Aside from that, you can also describe the form as entire, undulate, filiform, curled, or lobate. A very good one also would be to describe them based on their elevation. Okay, so of course, since it's an elevation, it should be determined by tilting the culture plate and looking at the side of the colony. So you can have a better view as to what is the elevation of the bacteria or the colony. So they can either be described as raised, convex, or dome-like. So this is definitely the more common elevation of most organisms. So convex or dome-shaped or dome-like, flat, okay, umbilicate or depressed at the center. No? So medyo there is a depression or a cavity at the center. Or pwede rin namang umbonate. So it's the opposite. Instead of a depression or instead of a hole or cavity, meron siyang bulging center. Or the center is bulging. Okay, so here we can observe some elevation. So for figure 8.10, these are diphtheroid colony with the appearance in an umbonate center growing on sheep blood agar. Okay, so diba meron parang bulge at the center. That's why we describe them as umbonate colonies. How about for figure 8.18? So this is also an umbonate colony. Fuzzy edge. But as you can see, medyo empty yung mga part ng colonies. Okay, so because this is a very specific characteristic unique to the organism that have grown. Okay, so diba parang may mga empty spots and they refer to it as pitting or pit. Kasi nga parang may empty spots or parang may nag-big onto it, diba? Pitting, ganun. So this is a very specific characteristic exhibited by Echinella corrodens. So that's why it's called corrodens or named as corrodens kasi nga it has a tendency to have this kind of growth. May mga pit or may mga craters, no, or mga empty spaces or empty spots, and we refer to it as pitting. So if may nakita kang description that says pitted agar, then that is most likely Echinella corrodens. Elevation. Okay. So here we have another examples of elevation or colony illustrating different types of elevation. Here we have Streptococcus pneumoniae. So it has an umbilicate elevation. So medyo may empty spots or may cavity or depression at the center. Okay? It can be in two forms. Pwede yung umbilicate, ganyan. May umbilicate or pwede rin naman yung penny edge. Okay? So, sometimes, so if you hear the description penny-like growth, then that is a characteristic of Streptococcus pneumoniae. Streptococcus pneumoniae kasi is capable of producing what we call the staphylokinase, where it can lyse some part of its colony. Kaya, yeah. No, not staphylokinase, sorry. It's not staphylokinase, but it has an enzyme that is capable of lysing the colony. Lalo na kapag older, no? Kapag older yung colonies, kaya medyo, kasi it grows normally, pero kapag medyo naging old na siya, we can observe this kind of um, growth. Okay, so penny-like growth is very characteristic of Streptococcus pneumoniae. How about for hemolytic viridans or alpha hemolytic viridans streptococci? They have an umbonate center and have a translucent grayer rough margin with an umbonate center. Another one, you can also use density. So how transparent, opaque ba siya, transparent, translucent, etc. etc. So for this, you can use light actually or the trans-illumination process para mas makita mo ng maayos. So again, kapag wala kang makita or very vivid yung color or very complete yung color, we refer to this as opaque. Okay? So it completely blocks off light. Translucent, and then we have transparent. Translucent, medyo may opaqueness ng konti, but it still allows the passing of light, therefore translucent na itsura. Parang semi-transparent, parang ganon. And if it completely allows light to pass through, then that is a transparent colony. So tignan glass, no, magkaiba ang transparent and translucent. So translucent, again, still blocks off a relative amount of light, but for transparent, it completely allows the light to pass through. Kaya transparent yung nakikita natin. Okay. Next is color. So again, this is also a very very good way to describe colony growth. So color is a term used to describe a particular genus in general. So colonies may be white, gray, yellow, or buff. So pinaka-common na color nila is white or cream. White colonies or cream. May iba din grayish. So for this, yan. So figure 8.13 illustrates a typical atypical colonies or a typical way as to most colonies look like. Yun, very typical colonies. So creamy white lang siya. And very common growth of colonies. And we have pigment production. Take note class, magkaiba ang pigment production and color. Kasi yung color, that is true to all organism. Okay? That is true to all organism. But for pigment, only a very select few can actually create a pigment. Okay? So pigment production is an inherent characteristic of a specific organism confined generally to a colony. Although some pigments will diffuse through the culture medium. Again, for color, lahat ng colonies na tutubo, they have a specific color. But for pigment, only a select few organisms can actually produce a pigment. And again, this is a very definitive or a very good way of identifying an organism. Kasi nga, iilan lang yung may kayang mag-produce ng pigment. So a very, very good example of that would be in the case of Serratia marcescens. So this is a bacteria or bacterium that is capable of producing a brick red pigment, as evident on the right culture media. So we can observe red pigment production, and this is a very good indication that the organism is Serratia marcescens, because it's able to produce that brick red pigment. So this is actually due to the substance called prodigiacin or prodigiocin. Hindi na kasya. Okay. So this is in part due to the substance which we call prodigiacin or prodigiocin, whatever you may prefer it pronouncing. This allows the serratia to be able to produce a red pigment. So just a little bit of information. So you might have heard news stories from other countries or even in the Philippines or in yung mga religious uh, statue, or how do you call that? Uh, so yung mga religious statue, I'm blanking. My mind is blanking right now for a better term, but you get my point. Yung mga religious statues, or even yung smaller ones. I forgot how you call them again. So, diba, you may have heard uh, anecdotes as to my instance daw na parang they cried red tears, parang ganun, diba? May mga instance daw somewhere wherein may lumalabas daw na red na dugo sa eyes ng mga statue. Okay? And a very good way to explain this is actually due to serratia marcescens. Okay? So, um, na-invade or nakapagtubo lang yung serratia marcescens doon and nakapag-produce siya ng red pigment on the statue, and that's why it gives us that bloody tears appearance. Okay? So, not sure what country where they actually did bacterial culture for this and identify that it's serratia, but that is a explanation, or that's one explanation as to how that happened. Okay? So, because of the brick red pigment of serratia marcescens. Also, take note, class, incubation at room temp actually enhances brick red pigmentation. Or this is very true, eh? Incubating the colonies in a higher temperature, just like I don't know, lower temperature, room temperature, sorry. Incubating the colonies at room temperature, specifically for serratia, enhances the production of the pigment. Ayan. Pigment production is generally enhanced by growing the organism at room temperature. So between 20 to 24 degrees Celsius. So there are also other types, orong spelling pa yung Pseudomonas natin. There are also other types of organism that produce very characteristic pigment. So we have one aside from Serratia, we have Pseudomonas. Okay, so Pseudomonas, or the genus of Pseudomonas, can actually produce a myriad of different types of pigment. So they have, they are capable of producing, or they have specific substances that allows them to do so. But only Pseudomonas aeruginosa can have that greenish blue pigment because of thiocyanine. Kasi yung ibang Pseudomonas pwedeng red, brown, ganyan. Iba rin pag gumagamit tayo ng UV light. That's a different approach, class. If you use a woods lamp, which is a UV lamp, it gives off the different color. Iba na naman yun. Just like si Prevotella, I think. Or si Vilonella, mga ganun. So, iba yung natural pigment and iba yung color produced when they are exposed to a woods lamp. Medyo magkaiba din yan. There are some organisms that have different colors depending as to where you're going to be observing it. But anyway, let's focus on what we can readily observe. So right from the get-go, kapag paglabas mo ng incubator, makita mo na siya. No need to use anything else. And that is what we are discussing right now. So, balik tayo kay Pseudomonas. Again, all Pseudomonas are actually capable of producing a specific type of pigment. Usually, very common is brown and red. But only Pseudomonas aeruginosa can produce that characteristically bluish green or greenish blue pigment due to the presence of thiocyanine. And also, it may sometimes look metallic. So, metallic green colonies, Pseudomonas aeruginosa. So, Serratia, similarly, Rubidea as well is capable of producing brick red colonies, especially at room temperature. Then we have Cloivera, Chromobacterium violaceum, na no need to memorize, while its color would be practically in the name, or it's named particularly due to that characteristic of a purple pigment. And lastly, we have Prevotella melaninogenica that produces a brown-black pigment, similarly named after the pigment that it produces. So we also have the consistency or the texture. So we can determine this by using an inoculating loop. So kawakan mo lang using the or yeah. So yes, observe or touch the colonies using a sterile loop, and you can describe it as maybe brittle, creamy, waxy, or dry. Yeah, so may mga consistencies and odor. Ito ulit. So aside, yung mga very definitive characteristic natin would include pigment production and odor. Okay, so, per my personal experiences, para-pareho lang ng amoy nila. Para silang mabaho. I haven't actually encountered wherein there's a specific or distinct smell as to what different organism. I've dealt with a lot of different types of organisms din naman during my duties before, pero they all smell the same to me. Okay, but apparently, as per our microbiologist's observation and as our textbook says, odor can also come in different forms depending on the type of organism grown. So but take note last safety precautions, okay? Odor should only be determined when the lid of the culture is removed and the odor dissipates into the surrounding environment. So what you'll do is going to be opening the lid slightly, ganyan, and wait for the odor to dissipate into the surrounding environment. Hindi mo diretso singhotin yung culture media, okay? Kasi again, that's their risky move. No, do not put a colony anywhere near your face. Again, you can get infected or yeah, may pwede magkaroon ng contamination if you're going to do that. So again, practice safety. Yeah, practice safety. And if you're going to determine the odor, do that by just opening the lid and let the odor dissipate within the room. Okay, do not directly sniff it. So what are some of the characteristic odor? Sabi nila, Staphylococcus aureus have that old sack smell. Pseudomonas naman have that fruity or grape-like smell, okay, because it has a specific substance, like 2-aminotryptine, something like that, that allows it to create that fruity grape-like odor. For Proteus mirabilis or Proteus species in general, they have that putrid burnt chocolate odor. And for Haemophilus, mousy, ganun daw yung smell niya. Amoy mouse or mousy or mouse mess. And we also have Nocardia, which smells like a freshly plowed field. Maraming type of odor associated with specific organism class. We also have Echinella corrodens, having bleach-like smell, odor, sabi ng mga references, and many others. So I've only enumerated some of the very common ones. And you can also use hemolytic pattern. So using blood agar, hemolysis can be observed surrounding the colony. Okay, this is actually due to the ability of the organism to produce very specific enzyme. And depending on what enzyme they can produce, it correlates with what type of hemolysis we can observe. So let's talk about hemolytic pattern later. And for hemolytic pattern, you can also use the transillumination. So basically, tatapat mo lang yung culture media sa may light, no, para mas ma-observe mo ng maayos yung hemolysis. Then other characteristics such as combination of different types of appearance. Then broth. Okay, in broth culture class, the positive way of identifying organism is through turbidity. Okay, that's kung sa solid agar, you can say that there is growth through the presence of actual colonies, solid colonies. For broth culture, you can say that there has been growth through turbidity. Pag turbid yung culture, may growth. Okay? So, meaning kung may mga natago kayo dyan na tubig or natago kayo dyan na any beverage that you forgot to drink, it's hidden somewhere, pag turbid na yan, most likely there's have been already bacterial or any type of um, pathogenic or microbiological growth within that. So, tapan mo na. And i-dispose mo properly. Kaya mo i-dispose lang somewhere kasi baka may something na lalo na kapag fungi yung tumubo. Kasi again, spores of fungi are very easily disseminated. Same lang din nun pala, class. Kapag contaminated yung plate, tapos may mga fuzzy part, huwag mo na open yan. Diretso, i-decontaminate mo na siya agad. Okay? Put some either bleach or acid. So I recently learned in Norma, they use acid, very strong acid to decontaminize their culture. So yun, just warning, no, kapag may mga ganong instances, since sobrang fluffy na huwag mo na siyang i-open kasi all spores are easily disseminated. Again, so here we have examples on how we can evaluate the colonies. So according to their shape, margin, density, color, pigment, consistency, etc., etc. So in here, the colony are irregular. Ayan. So this growth is actually very characteristic of Mycobacterium tuberculosis. Ang description pa nga nila dyan, kwan daw, cauliflower colonies. Baka ba yung cauliflower? Pwede na, pero not quite. But again, this is the growth of Mycobacterium tuberculosis. And we also have another one here, so circular, smooth yung shape or yung margin, density is opaque, elevation is convex or dome-like, color and pigment, or as a initial identity, we can identify this as Serratia because of the red pigment. But again, there are organisms that are capable of producing other red pigments, so initial muna na finding as of now. And we also have here, this is again a very typical growth of colony. So yung I have here very similar, but ang difference is the production of pigment or color. Okay, this is a color of there is a specific color of the colony. This is golden or yellow. So what do you think is the organism that is capable of producing golden colonies? That is Staphylococcus aureus, particularly on MSA agar or MSA, redundant, MSA or the mannitol salt agar. So kaya nga it's called Staphylococcus aureus from the old name of gold, which is aurum, because of its characteristic golden color when grown on mannitol salt agar. Yung mga fluffy colonies or very filamentous colonies. Here we also have a typical colony then, but this time there is hemolysis. Okay, there is a hemolytic pattern. Yun yung nakikita natin na parang halo effect surrounding the organism, that is the hemolytic pattern. Ito naman, flat, di ba? A very similar characteristic, but in this one, flat siya, hindi siya dome-like. And that is how we evaluate colonies. In real life, this is what our textbook says, class. But in real life setting, since I've already told you earlier, from previous discussion, karamihan kasi ng laboratory, they already use automation or machines to identify the organism. So, what they usually describe, commonly na lang na sinasabi is size or not even, size, color. Yun, size and color na lang yung ginagamit nila. So for example, small circular creamy colonies. Ganun na lang. na very basic. Kung ano yung makikita mo, yun na lang talaga. Hindi na nila describe si the density, si margin, si elevation. They just simply tell what they can readily observe. And again, that includes the color and the size of the colonies. Or sometimes they also tell us na, ayun, convex colonies, ganyan. Depending din naman sa SOP ni laboratory. But again, they only describe what they can readily tell. No need to identify the margin, mga ganyan, consistency, etc., etc. Okay, how about last here? What can we tell or what would be the characteristic of the organism pag medyo mucoid? If you can still remember, pag medyo mucoid yung colonies or basa-basa, that is that is an indication that the organism is capable of producing capsule. So they're actually using the sugars that are present in the um, culture media to be able to produce their cups because they are made out of a very structured layer of polysaccharide. And that's why we can observe a very mucoid or parang basa, no? Almost like watery, glistening type of growth because ginagamit nila yung sugars to essentially create their capsule. So that's it for evaluation of bacterial colony growth. Now let's talk about the very specific. So after learning the textbook way of evaluating colonies, let's talk about the very specific color or very specific growth that we can expect to observe on different type of culture media. So ito, generally for everything, but next, we are going to be talking about the culture media in general. Yun. So I did not discuss this part yet because I want it to discuss after na natin ma-discuss yung evaluation. So we've talked about FT or fluid thioglycolate. Though one thing I did not discuss yet is yung growth. Okay. So depending on their growth class, you can actually identify their environmental or oxygen requirement. By the way, I think I wasn't able to mention this. Kapag before mo inoculate, you need to boil it to, again, um, sterilize the culture, essentially. Again, to sterilize the culture, another way of making sure that there is no contamination, iboboil mo muna yung culture. So how do we do that? Kuha ka lang ng test tube holder. Tapos ipitin mo yung culture bottle, tapos i-heat mo until may mag-bubbles na mag-form. Suddenly magbo-boil siya or magsisimmer siya. Pag may nag-boil na, then you can keep it out of the flame, wait for it to cool down, and you can use it. But boil it first before mo siyang gamitin. Kasi, I've mentioned earlier that turbidity is an indication na there is bacterial growth on a culture or on a broth or in a broth culture. But there is a very specific threshold kasi. Or there is a specific threshold of organism growth that we can observe. Meaning, meron lang specific amount of bacteria yung kayang that makes things clear. May specific amount lang ng bacteria wherein magkakaroon ng turbidity. Okay? Not because clear sa paningin mo yung broth, that doesn't 100% ensure us na wala ng bacteria doon. Kasi merong specific amount lang ng bacteria na yun lang yung point na kayang makita ng eyes natin na may bacteria. Okay? Or there is only a very specific amount of bacteria that will provide a very observable turbidity. Okay? Hope you get it by now. And yung point lang na yun, yung pwede natin makita. But if it goes beyond or it goes below that specific threshold, hindi na natin siya makikita using our naked eye. But there is still bacterial growth on that. Therefore, to prevent any mistakes i-boil muna natin yung mga broth natin before we use it for inoculation. Now let's go back to the fluid thioglycolate. If you remember, FT is fortified with thioglycolic acid, which is a reducer and will prevent uh, excessive amount of oxygen from being stored or from being present within the culture. So because of this, we can actually identify the oxygen requirement based on their growth. So letter A, una, i-describe muna natin yung growth nila one by one, and then let's try to see what would be the meaning of it. So for letter A, diba, as you can see, there have been growth all throughout the broth. Again, there have been growth all throughout the broth. Diba? As you can see, my turbidity all throughout the broth. Okay, so for letter B, may flocculation. The one here is my flocculation. As you can see, yung mga parang nag-clump, so that is referred to as flocculation. For letter C, most of the growth is confined on the upper portion of the tube. And for letter D, opposite naman, most is confined at the bottom portion of the tube. Now, what would be the meaning of these? So, for letter A, or mas madali na lang sa letter D. Sa letter D, ang growth lang is at the bottom. And this means that this is the growth of anaerobic organism. So, bakit nangyari to? Kasi nga, let's take a look at the tube. Pag close mo nung tube na yon, where do you think, what area do you think has more oxygen? Is it the top or the bottom? Of course, it's the top, diba? Kasi it's nearer to the mouth of the tube, it's nearer towards the outside environment. Kaya of course, it would make sense na mas madaming matitirang oxygen doon. Therefore, if the organism is anaerobic, it will tend to grow at the bottom wherein there is little to no oxygen. Okay, it would prefer or tend to grow at the bottom because it has smaller amount of oxygen. It has better chances of growing there because of the lack of oxygen. How about for letter C? The complete opposite. Nag-grow siya on the upper portion of the culture media because it is more concentrated with oxygen or mas maraming amount of oxygen. Therefore, this is a aerobic or this is an aerobic organism, an space aerobic organism. And for letter A, since it was able to grow all over, wala siyang pakialam kung merong oxygen or wala, this is a facultatively anaerobic organism. Okay, blood agar plate. What identity or what description characteristic can we tell using blood agar plate? It is the hemolysis. So, pwedeng beta, alpha, or gamma. And this is usually used to describe or differentiate what type of organism. It's the Streptococcus, diba? Streptococcus pneumoniae, Streptococcus pyogenes, and we have Agalactiae. So for beta, we refer to it as complete hemolysis. Okay, complete hemolysis. As you can see, there is a very large zone of clearing through the streak pattern, di ba? Kung saan ka nag-streak, syempre doon tutubo yung organism and doon ma-observe yung hemolytic pattern, di ba? With the sides of the colony. So in here, you can observe beta hemolysis. Again, this is complete hemolysis. Therefore, you can see a large clearing within the sides of the colony. How about for alpha? For alpha, we refer to as incomplete or partial hemolysis. Because as you can see, there is a small amount of clearing within the sides of the colony, but not big enough. Or it's not as big or not as clear and definitive as the one in beta. Because for alpha kasi, yun, incomplete or partial lang yung hemolysis. The enzyme is not strong enough to actually completely lyse or destroy the red blood cells. Therefore, hindi masyadong mag hindi masyadong clear, hindi masyadong malaki yung zone of hemolysis. Aside from that, alpha hemolysis tend to have a green color because of the remaining unlysed hemoglobin being transformed to um, urobilin or biliverdin, sorry. For the alpha hemolysis, it also has a tendency to have a greenish color because the remaining hemoglobin uh, converts into biliverdin. Kaya nagiging greenish yung color. And lastly, gamma, wala kang ma-observe na clearing at all. So no hemolysis, we refer that as gamma hemolysis. So yung mga signs na pattern, yun yung tinatawag nating hemolysis. So we can have further identity or further clues as to what is the identity of the organism using sheep blood agar by identifying their hemolytic pattern. Another one, Makoti. Aside from being able to grow gram-negative organism, it can also tell us a very specific characteristic, and that is the ability of an organism to ferment lactose. And this is a very good way of differentiating the family of Enterobacteriaceae. I'm not sure if kung nasan na kayo sa lecture, but sakit ng ulo Enterobacteriaceae because it's a very large family. And is composed of a lot of different organisms, and these are differentiated through it is mostly differentiated through their ability to ferment lactose. So kung nakakapag-ferment sila ng lactose, they would have pink colonies. But for negative, they would have clear or yellowish colonies. Actually, ang mag-yellow is yung agar. But yung color ng colonies are clear or translucent. Okay. Translucent colonies or clear-looking colonies, those are the characteristic of non-lactose fermenting colony. So what would happen? Or why is it we're able to observe this kind of reaction? Because of the pH indicator, which is neutral red. Diba, as I've mentioned, if there is a pH indicator or color indicator, there would be a change in color depending on the pH of the culture media or the environment. When lactose is fermented plus, it leads to the production of sugars or any carbohydrates in general. Sugar, sorry. Any sugar or any carbohydrate, if they are fermented, this leads to the production of acids or acid products. And of course, ano bang pH ng acid? Diba? They are very low. And once the lactose is fermented and once acids are built up through the medium, syempre yung pH mag-change. And it will tell us if the organism is a lactose fermenter or a non-lactose fermenter. At a lower pH, at around 4.4, a neutral red assumes a red color or a pinkish color, as you can see from the image. Therefore, if nakapag-produce ng acids yung organism through the fermentation of lactose, mag-shift yung pH to a lower pH. Therefore, makikita natin yung pinkish or reddish color of colony. So if you're going to differentiate, diba ito yung regular na color ni McConkie, tapos meron magkakaroon ng a deeper pink color versus non-lactose fermenters, which will have a more alkaline pH because of the absence of acid production and will assume yellow color agar with clear colonies. So ano yung mga example ng lactose fermenters natin? Very good example is favorite si Escherichia coli. Escherichia coli, Klebsiella pneumoniae, and sa Enterobacter. Those are very good examples of lactose fermenting organism. Actually, yun yung tinatawag nating rapid lactose fermenters. Not lang yun, may refer to this as the eke. Yung rapid, meaning mabilis silang mag-ferment ng lactose. Tatlo lang sila class. So, madali lang silang i-memorize. The rapid lactose fermenters of the family of Enterobacteriaceae belong to these three genera. We have Enterobacter, Klebsiella, and Escherichia. Chocolate agar, walang masyadong growth yan. We also have phenyl ethyl alcohol, which is a sheep blood agar supplemented with phenyl ethyl alcohol that is used to inhibit the growth of gram-negative bacteria. So kung meron tayong McConkey that inhibits the growth of gram-positive organism to promote the growth of gram-negative ones. We also have PAE, which does the opposite. It's for gram-positive organism because it's fortified with phenyl ethyl alcohol that inhibits or suppresses the growth of gram-negative bacteria. Okay, let's now move on to a more specific type of culture media. We have Thayer Martin, and all of this are used for the growth of gonococci. We have Neisseria gonorrhoeae. So, as you have already mentioned, or as you may have already know, Neisseria gonorrhoeae is a very fastidious organism. Maapesi, Neisseria gonorrhoeae. Therefore, we need to culture it within the best or the most optimum environment as possible. And this is achieved through the use of very specific culture media that is presented on the given slide. So, maapat siya. Thayer Martin, modified Thayer Martin, Martin Lewis, and New York City agar. So, these are very selective agar kasi meron siyang inhibitory agent against a lot of possible organism that can compete with the growth of Neisseria gonorrhoeae. Siyempre, medyo weak si gonococci kasi marami siyang requirement para makapag-grow. If hindi maganda yung environment, uunahan siya ng mga contaminants in growing or in other organism. Uunahan siya sa pag-grow. Kaya kailangan natin ng very selective medium in the presence of these type of culture media. So, again, merong type of inhibitor for each type of organism. So, for gram-positive, vancomycin. For gram-negative organism, colistin. Okay? Para-pareho yan. Kahit na anong types of media, their difference would be within their antifungal agent. Si Thayer Martin uses nystatin. Similarly, with modified Thayer Martin, their difference is kay Proteus. Okay, so bakit pine-prevent niya specifically yung growth ni Proteus? Kasi yung swarming phenomenon, diba? That can inhibit again the growth of the organism. To prevent the swarming, the growth of Proteus, also preventing the swarming phenomenon, ilalagyan nila ng antibiotics specifically against Proteus species. So for Thayer Martin, originally, wala siyang anti-Proteus agent, but the modified one contains trimethoprim lactate. For Martin Lewis and New York City agar, pare-pareho na sila ng agent against Proteus. Their main difference would lie on the antifungal. So Martin Lewis uses anisomycin and New York City agar uses amphotericin B. Again, take note that these are all antibiotic. And they're also differentiate within their agar base. Thayer Martin, cap, and for New York City agar, ang agar base na is peptone. Then we also have XLD or the silene lysine deoxycholate or desoxycholate agar. So this is also a very selective and a differential media, primarily for the Shigella species and Salmonella species. So this particular culture media inhibits gram-positive. So makes sense kasi nga pinopromote niya yung growth ni Shigella and Salmonella, which are gram-negative rods that belong to the family of Enterobacteriaceae. So it contains a phenol red indicator which detects acid production and also detects hydrogen sulfide production. Now let's see what can we expect. Ang daming one. What can we expect when it comes to the growth of organism on a xylose lysine desoxycholate agar. So if we're going to be differentiating Salmonella and Shigella, these are their expected results. Si Shigella, clear colonies, and Salmonella, clear colonies with black center. So how does this happen? Ano ba nangyayari? So they both have clear colonies because they are both non-lactose fermenter. Okay? They have a clear colony because they are both non-lactose fermenter. And bakit may black center sa Salmonella? Bakit siya may black center? Kasi it's capable of producing H2S or hydrogen sulfide. The way we can tell if an organism is capable of producing hydrogen sulfide is through the observation of black colonies. And that is exemplified here by Salmonella. So both Shigella and Salmonella are non-lactose fermenting bacteria, but only Salmonella is capable of producing hydrogen sulfide, therefore it has that characteristic black colonies or black center. Actually, other family or other members of Enterobacteriaceae can actually grow on XOD and they have yellow. Yeah, kapag non-lactose fermenter, clear lang din yung color. But kapag um, for a lactose fermenter, mag-yellow yung agar. Another one, we also have hectone enteric agar, and this is also used for the differentiation of Salmonella and Shigella. So it uses bromthymol blue and acid fuchsin as its color indicator. How about for hectone enteric agar? Ang difference naman, pwedeng so mag-change yung color from green to red depending on your ability to ferment lactose again. Kasi nga, if we're talking about Enterobacteriaceae, one of their defining features would be whether they are able to ferment lactose or not. Kaya parating, ganun ulit yung differentiation. So, kapag mag-stay na green or clear yung agar with clear colonies, just like in here, again, this is a non-lactose fermenting organism. Pero kung mag-red siya, then that is a bacteria that is capable of fermenting lactose. Now, balik tayo on the differentiation of Shigella and Salmonella. Very similar reaction on the XLD. Both are clear, but only Salmonella that produces H2S has a black center. We also have eosin methylene blue. So eosin methylene blue inhibits the growth of gram-positive organisms. So EMB is also used for the growth of gram-negative organism, particularly the family of Enterobacteriaceae. But EMB is a very, very good media to cultivate E. coli or Escherichia coli because it has that characteristically green metallic sheen. Kapag nakabasa na kayo ng green metallic sheen class on EMB, take note, EMB, EMB, green metallic sheen, that is very, very characteristic of Escherichia coli. Then, green metallic sheen on EMB, automatic Escherichia coli. Meron pa kasing kayang mag-produce ng green metallic sheen. If you remember, sino yung isa? Si Pseudomonas aeruginosa, but not on eosin methylene blue. For selective culture media, then specifically for Pseudomonas aeruginosa, we have cetrimide, for example. Pero kung specifically eosin methylene blue, green metallic sheen, matic, Escherichia coli. And you have TCBS, or thiosulfate bile salt sucrose, and this is for the isolation. It's a very selective medium for the isolation of the Vibrio cholerae because of the presence of thiosulfate, bile, and salt sucrose. Kasi thiosulfate bile, specifically bile, inhibits the growth of most organisms, and it has um, thiosulfate and sodium citrate that maintains the alkalinity of the medium. So the alkalinity of this inhibits the growth of Enterobacteriaceae, and it's also a very good environment for Vibrio species, specifically Vibrio cholerae, to grow in. Gusto nila ng mas alkaline na environment. Therefore, if you hear alkaline peptone water, that is also for the growth of Vibrio species, particularly for Vibrio cholerae. And take note, may sucrose na naman kasi some species of Vibrio are capable of fermenting sucrose. So here, it has, again, it has a color indicator because the color can change depending on the ability of the organism to ferment sucrose. And the pH indicator are bromthymol blue and thymol blue. So the acidification of the medium results from the fermentation of Vibrio. So kung yung Vibrio, kaya niyang mag-ferment ng sucrose, it will turn yellow. If not, it will remain green. So, an example of that, ano ba? I think Vibrio cholerae and Vibrio mimicus. No. I'm gonna start sa that. Vibrio, kalimutan ko na. Pero sigurado ako kay Vibrio cholerae. Let's get back to you on that. But one very good example of a Vibrio species that is capable of fermenting sucrose is Vibrio cholerae. Then you also have buffered charcoal yeast extract. So this is a media that contains charcoal which acts as a detoxicant, and activated charcoal decomposes hydrogen peroxide, a metabolic product toxic to Legionella species. So we have here a very specific type of culture media that again selects the growth of a specific organism through the addition of a specific substance. So in here, we have the presence of charcoal, kaya black yung color niya. So anong ginagawa ni charcoal? Testify it's a detoxicant that decomposes hydrogen peroxide. So hydrogen peroxide is an example of a reactive oxygen species, and this is a metabolic product that is toxic to Legionella. So Legionella is grown on buffered charcoal yeast extract. So the yeast extract acts as a rich source of vitamins again to further promote the growth of Legionella. Legionella pneumophilia. And these are grown on buffered charcoal yeast extract. Then we also have Lowenstein Jensen agar, and this is a glycerated egg-based medium especially used for culture of Mycobacterium species. So, underline natin sa egg-based. Bakit may egg-based culture media tayo? Kasi meron sa egg, it's a rich source of protein. And Mycobacterium species needs a higher concentration of protein to grow. Wow, bodybuilder pala si Mycobacterium species. Again, they need higher amounts of protein in the culture media in order for them to grow. Therefore, for some types of medium for Mycobacterium tuberculosis, they are egg-based. And for this, Congo red and malachite green is incorporated in order to inhibit unwanted bacteria. So similarly, with other fastidious organisms, mahirap tumubo si Mycobacterium class. Okay? They are slow growers. Specifically, Mycobacterium tuberculosis, they are slow growers. If we're going to be culturing them using the traditional method, it would take them at least a month to grow. Kasi nga sobrang slow grower sila. You will have to wait 2 months before you discard the culture media kasi nga slow grower sila. And this is not cultured, although gold standard yung culture for identification of Mycobacterium species, hindi natin siya ginagawa ngayon. It's very disadvantageous, no? Sabi, what if lumala yung TB ng patient, patay na siya, hindi pa tumatubo yung bacteria, diba? That can happen. So, we have more sophisticated and faster way of identifying TB or diagnosing TB right now instead of the usual culture media. So, hindi natin masyadong kino-culture si Mycobacterium tuberculosis. And to again promote the growth using the LJ agar, it's fortified with Congo red and malachite green, specifically with malachite green. Kasi lahat ng mga culture media for Mycobacterium has malachite green as an inhibitor to prevent the growth of any unwanted bacteria. So ito naman yung other one. If meron tayong egg-based, meron din tayong protein-based na gel. And this is illustrated through the Middlebrook agar. So it also contains a variety of supplements that will further promote the growth of Mycobacterium. So it contains albumin, glycerol, oleic acid, and low amounts of malachite green as an inhibitor. Okay. Then we also have a biphasic medium. So as you can see here, it has two phases. Okay, meron siyang solid and liquid part. Okay, it's called a biphasic medium. It has both a liquid and a solid component, and this is actually utilized to grow highly pathogenic organism. Bakit? Siyempre sa dalawang Sa isang culture, nandun na yung dalawang type, solid and liquid, so that you don't have to inoculate separate ones. And this is used for highly contagious or highly pathogenic organisms. So a very good example of this would be the Castaneda medium used to grow Brucella Brucella. At I think bioterrorism agent level A itong mga to kasi nga madali siyang ma-spread. And the symptoms are actually very severe as well. So these are organisms or bacteria that comes from animals usually. So zoonotic ang transmission of Brucella. And again, yung marinig yung mga bioterrorism agent A, B, they are classified depending on how they can easily be transferred or transmitted. So A, highly transmissible. B, transmissible but not as transmissible as A. Ganyan. And I think A, bioterrorism. I think B at B, bioterrorism agent. A, I think A si Brucella, B si Francisella, A din si anthrax. Okay? So, again, the very reason why we have a biphasic medium, nandun na si solid and liquid para kung mag-inoculate ka, isa na lang. Sana na lang. Hindi mo na kailangan mag-open ng another one to prevent the transmission of the organism. Okay, and these are the other culture media that are available, complete with their components and other comments as well as for their primary purpose. So, makulay ang mundo ng microbiology class because of the a lot or because of the presence of different types of culture media, and with different types of culture media also the different type of growth. So as you have learned, my pink, my yellow, my green, my black, etc., etc. And again, all of these are very helpful towards the identification of the organism. Okay, so that ends our discussion on the evaluation of culture media.